Hi everyone, I am Yokimiko David-Torrejos, and today I'm going to share with you our study, which is entitled Transcriptome Mining for the Development of a Molecular-Based Nitrogen Sensor in Oryza sativa. The growth and development of the rice plant is dependent on the supply of nitrogen, which makes it a limiting factor in rice plant. It is a component of most metabolic and biological compounds such as nucleic acid, amino acid, and chlorophyll. It is also present in the soil in the form of ammonium, nitrate, and amino acid. When nitrogen is supplied at optimum concentration, it could promote a healthy and highly productive crop. However, when a plant suffers from nitrogen deficiency, it could ultimately lead into lesser productivity, similar with the oversupply of nitrogen. When nitrogen is supplied more than the recommended amount, they appear very green in color. However, they are very prone to insect and pathogen attacks. In addition to that, nitrogen that are not taken up by the plants is lost in the environment through volatilization and leaching, causing harm and damage to our environment. The proper management of nitrogen remains one of the challenges faced by our farmers and researchers. And through time, several technologies have been developed to measure nitrogen concentration, both in soil and in plants. And the most common method used by our farmers are the spad meter and the leaf color chart. In 2018, a molecular sensor was also introduced, which uses the internal response of the plant to monitor nitrogen. However, these methods are either expensive, destructive, or dependent on the delayed physiological response of the rice plant. Hence, a rapid and real-time monitoring of nitrogen is necessary. To better understand how plants respond to stress such as nitrogen deficiency, transcriptomic profile analysis like RNA sequencing can be done to acquire information on the dynamic expression profiles of the different genes and regulatory factors in response to a specific stress across a different time periods. The whole process of RNA sequencing can be divided into two general steps. The first part includes PCR amplification and sequencing, followed by the computational analysis of the sequencing reads. However, this process generates huge amount of data that we usually analyze using different softwares and programs. The identification of genes can be useful in developing transgenic rice plants that can adapt to nitrogen stress, or in developing molecular sensors to monitor nitrogen stress. Hence, in our study, we aim to identify genes that are highly responsive to nitrogen stress using their expression patterns in the presence and absence of nitrogen. Here, we integrated a screening method that served as an extension of the RNA sequencing pipeline to identify genes that behave oppositely to the presence and absence of nitrogen. The RNA sequencing data was provided to us by Professor Jukon Kim from the Seoul National University. The huge amount of data describes the transcriptomic response of the Oryza sativa cultivar Nippon Bare into two nitrogen conditions, starvation and adaptation. And presented here are 6 out of the 44,714 transcripts and their FPKM values for each of the sampling points under the two nitrogen condition. And here are the different sampling points for nitrogen starvation and nitrogen adaptation. Before screening, we initially removed transcripts with FPKM values that are less than 0.1 on both nitrogen condition. After data filtering, we were left with 33,616 transcripts from the leaf tissues and 35,110 transcripts from the root tissues. The screening method was based on the expression patterns of the transcripts during nitrogen starvation and adaptation. Here, we were able to generate a 4-numbered codes, which represents the expression pattern from the 5 sampling points. The code was designed by evaluating if there is an increase or decrease between adjacent sampling points, and we denote 1 for increase and 2 for decrease. For the characterization of genes, we perform subcellular localization analysis and gene ontology enrichment analysis to identify the dominant terms within the biological process molecular function, and cellular components that may be associated with nitrogen stress. Then the pathway analysis for these selected genes was performed using RiceFriend. Using the algorithm that we designed, we were able to generate 16 patterns that we assigned to pattern categories 1 to 16. The genes with similar trends are assigned to each pattern, which represents the molecular response of the rice seedlings to nitrogen starvation and adaptation. And since our aim is to identify genes that have a constant response to nitrogen, we selected those with pattern categories 1 and 16, which represents the upregulated and downregulated transcripts. And during nitrogen starvation, we also consider those that have a delayed response, and these includes pattern categories 8 and 9 under nitrogen starvation. Delayed response under adaptations were not included because of the short interval between 6 and 12 hours. And the distribution of the transcripts or genes using the pattern categories are presented in this graph. And here are the number of transcripts that were upregulated and downregulated. And to summarize, after data filtering, the number of transcripts were reduced to this number, and the numbers that corresponds to the regulated genes for each tissues under N- for nitrogen starvation and N+ for nitrogen adaptation. When these genes were plotted on a Venn diagram, there were 71 and 6 upregulated genes that are common to both leaf and root samples during nitrogen starvation and adaptation. The 71 upregulated genes common in leaf and root samples under nitrogen starvation were enriched with GO terms physiological process, cell death, and protein metabolism within the biological process. On the other hand, the 6 upregulated genes that were common to both parts during nitrogen adaptation were enriched with photosynthesis and carbohydrate metabolism. On the downregulated genes, there were 85 and 82 common genes in leaf and root samples under 2 nitrogen condition. From these genes, there were no enriched GO terms that were identified. We generated 2 general rules to help us in designing an algorithm, wherein we select only genes that show opposite expression on both nitrogen condition. For instance, we have here gene X and gene Y with their corresponding expression patterns and pattern categories. We designated 1 for upregulated, which includes pattern categories 1 and 9, and 2 for downregulated, which includes pattern categories 8 and 16. For a transcript to be selected, it must have an opposite expression to both nitrogen starvation and adaptation, similar with gene X here. Using our designed algorithm, we were able to identify 135 and 67 genes from the leaf and the root tissues. These genes correspond to the 0.40% of the genes on the leaf tissues and 0.19% of the genes from the root tissues. And these genes showed an opposite expression during nitrogen starvation and adaptation. The difference in number is also expected since root and shoot tissues or leaf tissues respond differently to nitrogen availability. When we conducted the GO enrichment analysis, there were 13 GO terms associated with the 135 genes found on the leaf tissues. Presented here on the table are the different GO terms and the number of genes associated with these terms. Dominant terms within the biological process were cellular process, physiological process, and cellular metabolism, which corresponds generally to the normal cellular, biological, and metabolic activities of the cell. Within the molecular function, binding and protein binding were the dominant terms. The subcellular localization analysis of these DEGs show their distribution in the different cell parts and organelles. When we examine the genes present in the cytosol and plasma membrane of the leaf tissues, genes found on the cytosols were enriched with response to stimulus, response to stress, and protein binding, whereas the genes present in the plasma membranes were enriched with transport, localization, and transporter activity. When plants respond to stress, such as nutrient limitation, their metabolic activities are altered by upregulating and downregulating various genes involved in stress response. Pathway analysis show the involvement of these genes in various biological and metabolic pathways. Some of the genes from the leaf tissues are involved in ubiquinone and other terpenoid quinone biosynthesis, oxidative phosphorylation, RNA degradation, glyoxylate and dicarboxylate metabolism, and carbon fixation in photosynthetic organisms. On the other hand, pathways associated from the genes in the root tissues were mostly degradation and metabolism of amino acids such as valine, leucine, isoleucine, beta-alanine, tryptophan, and lysine. And aside from these, purine, fatty acid, and butanoate metabolisms were also identified from these genes. The detection of these pathways validates the effect of nitrogen availability on carbon and nitrogen metabolism. Here is a process flow describing our screening method. Proper monitoring of nitrogen is an important aspect in the field of agriculture. Several approaches have already been introduced to yield healthy and highly productive crops. With the aid of transcriptomic studies, several genes have already been identified to be responsive to nitrogen stress. Such genes can be used to generate molecular sensors that can detect stress in rice plants, and it can also be used to create transgenic plants with improved properties and high resistance to nitrogen stress. In this study, a pattern-based clustering approach to identify genes from a pool of transcripts in RNA sequencing data was introduced. Further screening and characterization could provide more insights into the role of these genes in nitrogen metabolism.